Stanford University.
So it gives me great pleasure tonight to introduce actually one of our newest chairs at Stanford. Dr. Michael Snyder is the Stanford Asherman Professor and Chair of Genetics, and he is the Director of the Center of Genomics and Personalized Medicine. He actually just joined the faculty after leaving Yale in July, and so what a way to welcome him here. He got his PhD at Caltech and then did his postdoc here at Stanford, and he's a world's leader in functional genomics. And I think you cannot walk around or drive around the Bay Area these days without seeing the blimp up there asking us all to get our genes tested. And so tonight he's going to talk about genetics and personalized medicine. So Dr. Snyder.
Okay, well, it's really a great pleasure to be back here after 23 years, and I'm really having a blast once again. So I am going to talk about genomics and personalized medicine. I haven't seen the blimp. I'm probably the only person in Palo Alto who hasn't, apparently. And I will know I'll be looking for it this weekend. So this is an outline of the talk. I will give a brief introduction on genes, DNA, cells, these things I know you've had already. I just want to make sure everybody's on the same page, and if you're not, raise your hand and ask questions and that sort of thing. And then I want to talk about about sequencing genomes, what's been going on and what we might expect. And while we go through that, you might just think a little bit about whether you want to get your genome sequenced. And then I want to talk about the impact of genomics and genetics on personalized medicine and what we are seeing right now and what we might expect to come. And I'll just say a little bit at the end, Stanford is launching a new Center for Genomics and Personalized Medicine, and we hope to be significant players in this whole area. So just to give you a feel for what this talk is all about. So I think all of you know this. We start with from one cell and ultimately grow into something that has 50 to 100 trillion cells. And Arnold perhaps has more cells than the rest of us, or else they're bigger. I'm not sure which. But anyway, that's what happens. And along the way, we make many, many different cells. And it's hard to define exactly what a different type of cell is, but there's at least 200 basic types of cells and probably many, many more in our body, all carrying out very specialized functions. As you see here, we have nerve cells that help us think, and fat cells that store things for metabolism, and liver cells that do things. And then, of course, every now and then, some cells come out that go astray. Cancer cells is obviously one of the most significant ones, and these often look closest to the cells they actually derived from, but they certainly are malignant. Excuse me, and we'll talk about that in a little bit. So it's the DNA that's in us that determines what we are and determines what each of these cells are going to be. For the most part, nearly every cell has the exact same DNA. There are a few exceptions. And this programs these different cells, and it also then ultimately programs us. And we are different because of our DNA. And as you probably know, our DNA is housed in chromosomes. And we have 23 copies and 46 total chromosomes, 23 different kinds, times 2, one set from our mother and one set from our father, to give us 46 total chromosomes. The DNA, as you've heard from Gil and others, is made of 4 bases, or nucleotides as they're called. These are distinct chemical entities, and they're listed up there in big letters, A, T, C, and G. And they string along in a sequence and form a very distinct sequence. So a string of these nucleotides is called a sequence, and that becomes relevant as we talk about sequencing. And what's shown here is one strand of DNA, but DNA is normally double-stranded, meaning there's a sequence on one strand, and then you have what's called a complementary set of DNA or bases or nucleotides on the other strand. So there's a specific pairing, such as A's are with T's on the other strand, and T's are with A's, and C's are with G's, and G's are with C's. So ultimately, you get a 2-stranded structure, such as the one shown here, and it has a slight pitch called a helix. And that is, in fact, the structure of our double-stranded DNA that's in all of our cells. So the human genome, then, is the collection of all of these DNA strands. And we usually refer to one set of these chromosomes as comprising of the genome. So there's one copy, if you will, is 3 billion bases or 3 billion letters, as is often the case. And just to put this, what this would be like if you were to put this in phone books, this would be the equivalent of 200 phone books, 1,000 pages thick. So there's a lot of information here stored in our DNA, okay? And it's a fairly precise, for the most part, you'll see in a minute, our copies are relatively similar. They're not identical or we would all look the same, but they're quite similar from one to another. So not so long ago, there was an effort launched, the Human Genome Project. It started in 1985, and it was completed in 2003. And along the way, there was a draft of the sequence made, meaning they got a lot of it put together, but it was still in pieces. It was a massive effort. It involved 2,000 people, as you see here. I'm trying to get my arrow up here, 2,000 people. And the cost is, it's a little difficult to estimate, but it's somewhere on the order of $500 to $1 billion. The whole project, and counting technology development and things, cost about 3 billion bases. And it was often referred to as analogous to the Moon project, to send people to the Moon. This is the genetic equivalent, to sequence one copy of the genome. And this copy that was sequenced was actually a pool of people. They took a number of people, pulled their DNAs all together so that you would at the end identify one individual with this. And the sequence they came up with is called the reference genome. Okay, so that's the thing, the first genome that got sequenced out. It's 3 billion bases that refers to one copy, essentially, of each of our chromosomes, and it's called the reference genome. And that was, hailed as a huge milestone, and it has been a huge milestone in completing this project. Before this time, there were maybe a handful of genetic diseases that were mapped on the order of tens that were precisely mapped down to the exact gene, as you'll see in a minute. But after this project was completed, it was a very useful framework to be able, now we know hundreds of genes, although there's still many more to know. There are hundreds of genes that cause diseases that have been mapped quite precisely. So there's a big effort, once the genome was sequenced, to try and understand how do you get from one of these sequences, again, 3 billion bases of this, to form a human being. And equally important, we want to understand not only how you get from the DNA to form a person, but what happens when something goes wrong and you don't get quite what you were looking for, okay? And so we want to try and understand the genetic basis of this. And this is really clearly one of the biggest challenges in medicine today, is to identify the genetic lesions, ideally before they occur, or before they manifest themselves as problems, and then try and set out corrective action. That's clearly an ultimate goal of this whole area. So one of the critical parts of the genome, as you might imagine, are the genes. You've all heard the term genes before. There's on the order of 20 to 25,000 genes in the human genome. And you would think we'd know after we sequence the genome exactly where they all are and what they're all doing and such, but nothing could be further from the truth. We don't know exactly where all the genes are. We know where a lot of them are. We know it because we can compare our genome, believe it or not, with that of the mouse, and the parts that are conserved is one of the good ways of finding where the genes are. That's one of the ways in which you can do this. There's several. But their genes, they're scattered throughout our DNA, and it's even more challenging than you might think, because these genes, they're typically having 1,500 of these bases or letters, but they're actually broken into pieces that are fairly short. And it turns out they only comprise a small fraction. The parts that actually encode things that ultimately turn out to be the functional units, which are the genes actually make something called RNA, which in turn makes protein. And those final functional units, if you look at the genes that lead to those proteins, it's a small fraction of our total DNA. Having trouble getting the arrows up here. It's only 1.5% of the DNA. So there's a sea of these bases, and our genes within those are a very small part. And so finding those hasn't been so easy. We found a lot of them, but we don't always know where the ends are and things like that. And that's one of the big challenges going on. People are trying to define exactly where all the genes are in the human genome. It's one of the things our lab works on, in fact. One reason you want to do this is because the genes actually make, what genes are turned on determines what cells get made. So in a nerve cell, you'll have a set of genes turned on that make all the RNAs and the proteins you need for making a nerve cell. And in the muscle cells, you'll turn on, some of them will be the same, but a lot of them will be different. You'll turn on things that specifically lead to making muscles, and your fat cells make fat RNAs and proteins and so on and so forth. So these 200 different cell types each make their own sets of, each have a set of genes that get turned on to make specific proteins. And so really one of the goals then is not only trying to identify the genes, but to determine which ones are turned on in which cells, and that's one of the biggest challenges. Okay. Any questions about any of this so far? Yep?
Yeah, so here you're saying that the DNA in all the cells is exactly the same in the all of the body, only the difference between different cells is what is turned on and turned off.
Yes. So the question is, DNA is the same in all the cells, and it's only a matter which genes get turned on or off determines which cells become which types. And that's exactly right. Oh boy, there's a whole bunch. We'll start at the bottom and work our way up. Okay.
Are you going to tell us how they get turned on and off?
I wasn't going to, because I wanted to spend a little more time talking about why we're different from one another, why you don't look like the person sitting next to you. There are a set of proteins that I can give you a 2-minute synopsis. There are a set of proteins that will sit on in front of these genes, and then they're responsible for bringing in the machinery that turns on those genes. And there are about 1,500 of those proteins, they're called transcription factors, that are actually encoded in our human genome. And it's really quite interesting. Different sets of those transcription factors are turned on in different cells, and different combinations turn on different genes. And so if you love combinatorial controls and thinking about nice complex problems, this is the perfect one for you. And there are groups like ours. We have a huge project to actually try and figure out which of these controlling proteins is turning on every single gene in the human genome. We've invented some methods for doing this. And then it's a matter of slogging it through. The problem is, ideally, you want to look at all 1,500 transcription factors in every single cell type and in every single condition if you can. It's a big challenge. But there's a number of groups working on this. So that's called the regulatory code, to try and crack that. So we now know the genetic code. There's a big push to try and decipher the regulatory code. It's a great question. Yeah, one of you two, sure, either one. Futures.
Question. First of all, the chromosomes are composed of DNA.
Yes.
So the DNA is the same. So what makes one chromosome different from another?
Yeah, I didn't explain that very well. So there are 23 chromosomes. They actually have a different piece of DNA. They're not all the same. The 3 billion bases actually refers to the ensemble of DNA from all 23 chromosomes. So that 3 billion is broken into 23 pieces.
So the DNA is not one continuous.
That's correct, yeah. It's 23 pieces. Yep.
Oh, okay, great.
Yeah, and they're, as you might imagine, they're quite large then.
Right.
They're huge.
And then the other question.
By the way, it's a very interesting problem how you pack what's literally 2 meters of DNA into tiny little chromosomes. So they're really super packed to be able to do that. Yeah.
Can you make sure to repeat the question?
Oh, I'm sorry, yeah. All right, I'll do that going forward, sorry. Otherwise I got to backtrack too much, yeah.
Follow-up. In the human genome, they had 2,000 people, but each person had a different DNA sequence. So how did they arrive at one?
Well, they all weren't sequencing themselves, I'm happy to say. They were actually, they were working, there was this pool of DNA that was all put in the same tube. And then, no, from about 50 different people. They mixed that into one tube, and then they made, I wasn't going to get into details of this, there was a 2-step process, but they basically broke it into tiny little bits. It's an interesting, the first step was to break it into 1,000 piece bits and sequence those, and sequence them at random, and sequence them many of these fragments. And then, how many of you guys are computer types here? Oh, lots. So you'll love this. So what you do is you take those 1,000 bases and you see how they overlap. And your mission then is to assemble them all. And in a perfect world, if these were all unique sequences, you could just put them all together and you'd have the whole human genome sequence. The problem is it's not a perfect world. We have repeats in our genome. And so it means assembling this thing is, imagine a puzzle with lots of identical blue pieces that you can't tell apart. In a sea of things that you can tell apart, when you hit these blue pieces, you're kind of stuck. And so you need ways of actually assembling it. I won't get into that. I mean, it's a great question. It takes a bit of time. So that's how a lot of it was put together, and then there were ways of actually stitching. Things were put together into larger chunks, and then those chunks were stuck together.
Thank you.
Yeah?
Following up on your discussion of chromosomes, given that genes, DNA, become proteins, which become everything else, and chromosomes are different collections of different genes, what's the role of chromosomes?
The role of chromosomes, so chromosomes are both DNA plus protein, and they basically keep things packaged properly. So ultimately, your cells divide, and I think, did Gil tell you about cells dividing and stuff? I would have assumed so. Oh, I forgot to repeat the question, sorry. All right, so the question was, what's the role of chromosomes? And it really is, it's a packaging thing. Just like you go in the supermarket, you don't want the contents all spilled out. Could you imagine if we didn't have things in packages? That's just like a cell. You want to put things in their proper compartments. Yeah?
I guess my question is the other side of what comprises chromosomes. There's little discussion of the 98.5% of DNA that doesn't comprise of chromosomes, the junk DNA. Is there research being done on that to see if there's any utility?
Yeah, so the question is, what about this other 98.5%? So I said 1.5 encodes parts that will be coding for proteins. And there's a lot of other stuff, and that's a big mystery. We think a lot of it is this regulatory stuff. So remember what I said, these transcription factors bind? They actually bind to regulatory elements, and they're very hard to find and pick out just by computer algorithms and things. You really need specific experimental tests to find these things. And so we think a lot of it is doing that, but a lot of it, nobody knows what it does. And some people like to say, well, it's just there, and we can evolve that stuff. That is, we can get new genes coming out of that. So maybe many centuries from now, there'll be some new genes popping up and such. But the bottom line is we don't know what most of it does. And some of it's thought to maybe assist in packaging and things like that. You guys are asking great questions. Boy, this could take a while here, I guess. All right, we'll do a circle here. We'll come to you last. Okay, yeah, go ahead.
To find the genes, is there a transcriptor hanging out always by where they're located? So we will find the transcriptors and there's a gene next to it?
Most people think of it as the other way around. There's a gene, and then the transcription machinery comes in and expresses this gene. That's the term, expresses. Oh, I forgot. The question is transcriptors. I think I learned by now, wouldn't you? So the question is where he called it the transcript, where this transcription machinery is occurring. Is that where the gene is brought? Most people think it's the other way around, where the gene is, the transcription machinery is brought in, that it makes this RNA, and then actually the RNA goes, this is all done inside something called a nucleus. And then the RNA goes outside and gets turned in, gets interpreted into protein that way in a process called translation. Okay, yeah?
When we talk about sequence, does that refer to the helix and the nucleotides, which come first, and the combinations and going up and down the various rows and up to the bases then is?
Yeah, let me rephrase this if I can. So yeah, what do we mean by sequence? Sequence is really, it's this distinct set of bases in a very particular order, because those bases get interpreted. So those 3 billion bases, for the most part, are in a very specific order. Again, there are differences, and that's what makes us different. We'll talk about that in a minute. But for the most part, most of the bases are in a very specific order, and then as they get interpreted to make RNA and later protein, that's very, very critical so that they're making the same protein. So the word sequence refers to that specific arrangement. That's correct, yeah. So that was the hard part, figuring out this 3 billion bases and what order they were in. Everybody knew there were 4 of them. That was easy. How do you put them together into those 200 phone books? That was the hard part. Okay, one more, and then maybe I'll move on, and we'll see if we can catch up a little. Yeah, so why 23 chromosomes? It's a great question, and you may know that different organisms have different chromosomes. So, mouse and kangaroos and things, and there's some organisms that will have lots of little chromosomes. And nobody really knows why. Some of it may be evolutionary artifacts. That is, these chromosomes are broken up and then they stay that way. The reasons once they get set up a certain way it's hard for them to change very quickly. It's because it's very, very important ultimately that our chromosomes, when we make our kids, one set goes into the sperm and same with the egg, that you get one and only one copy of each chromosome. If you start getting what's called missegregation events, if an extra copy gets segregated improperly, then the consequences can be quite deleterious. Either that individual will not grow if you have an extra chromosome, or Down syndrome is the classic case where you have an extra copy of chromosome 21. So we don't know why there's that many. As I say, it may just be an evolutionary artifact, but it's you can't have too many screw-ups as you go through evolution. Okay, good. Well, great questions. I think that's a good sign, means you're all on board. All right, so we'll start here. Identical twins that come from the same egg, so an egg that has somehow split. They don't always happen, by the way, at the first division. It can happen later. Generally come out looking pretty much the same. I think we all know that, and that's because they have the exact same DNA. But what about two different people? So you, again, sitting next, how are you different from the person sitting next to you? And until not so long ago, it was always assumed that there were just these single base differences called single nucleotide polymorphisms. I'll spell this out on the next slide. That there would be rare differences between one individual, and that would make them different from the other. Okay, and so here's a picture downloaded from the web. This is some guy named Steve next to the basketball player Yao Ming. And if I were to ask you a few years ago, if you asked any scientist, what's the difference between Steve and Yao, everyone would have said, well, it's these single base differences, and the frequency of those is roughly 1 in every 1,200. That's roughly how many differences there are. And what that means is because we have 3 billion bases, now some of them are repeat, you'll see the math isn't exactly right, but there's about 4 million bases then that are different between you and the person sitting next to you. Sometimes these sit in these coding regions I mentioned in the genes, but more often than not, they're outside the genes, and they could still be affecting regulation and such. So this is, until recently, thought the major form of variation in people. Recently, though, several groups, I'd say a lab at Cold Spring Harbor named Michael Wigler, Charles Lee in Boston, and more recently our lab has actually found these other kinds of events going on. And this is a big surprise. There were hints of this before from a number of groups, but I think the extent of this turned out to be a huge surprise. And these are called structural variations. So these aren't these single base differences. These are big chunks of DNA that have either deleted, duplicated, or gotten rearranged, typically inverted, in the human genome. So if this is that so-called reference genome I mentioned, you have an order of DNA segments here, ABC. You can get a deletion where, say, B got removed. You can get usually it's a duplication, an extra copy, although you could have a novel sequence. That does happen. So you might get an extra copy, say, of B here. And then lastly, you can have an inversion in which two segments are flipped around relative to one another. So our lab, I won't get into the details of this, but we invented methods for being able to actually find this in large numbers of these in the human genome. And from that, we found that there's about 1,000 of these that are different between any individual and the reference genome. And if you add up the numbers of these, how many bases are affected, it's more bases than those single base differences I mentioned before. So in fact, we think this is the major cause of variation in humans. Not everyone agrees with that, by the way. The people who love SNPs will tell you they're the most important. But those of us who just like math say, well, there's 10 times as many bases affected by this, so there's going to be 10 times greater effect from these. I think common sense is likely to rule out in the end, but I better watch it. This is on YouTube, too. I'm in trouble. Okay. Anyway, we think that this will have a major form of variation, yeah. So it's not that SNPs aren't important. Don't get me wrong there. They are important, but we think this is going to be at least just as important. Yeah?
I want to get clear on the size of those. They're smaller than a gene and bigger than a single nucleotide?
Well, that's the thing. Typically in a definition, oh yeah, the question is, so the size of these things, how big are these? Are they smaller than a gene? Are they bigger than a gene? by definition, we call them 1,000 bases or larger, and a gene is on the order of about 1,500 bases, although it's broken into bits. So these are often at least maybe smaller in size, but they will hit genes, you'll see in a minute. But they can be incredibly large. Some of them are megabases in size, so over 1 million bases that will differ between individuals. So it's amazing that there are these big chunks of DNA that are out there that are different. Okay. Oh yeah, one in the back.
Yes, so in regards, I know identical twins, but everyone else is about 4 million differences. But what about siblings or cousins? Are there like sections where they match at all? Or?
Yeah, so since you get half your DNA from your mother and half your DNA from your father, as you might imagine, you share half of that with your siblings then, right? Because it's a random assortment of chromosomes. You don't get all of your chromosomes from your mother, you get half. Same with your father. So again, you'll typically share half with your siblings.
Would there be 4 million differences still?
No, then you would have half of that, generally. It's a good question. Yes?
Reference sequence, is it like the median? Because those two people had 4 million differences, and here you're having 1,000 with a reference. So is the reference taken in such a way that most people fit in within 1,000?
So the question is I'm not sure I understand the question. Try me one more time here.
Is the reference a hypothetical sequence?
No, the reference is a real sequence. It's a sequence that came out of sequencing these 50 people, okay? And that's what you're ultimately comparing against. So that's the thing we have sequenced. And now and this is important. You're actually touching on a part I was going to get to later, but when we actually when you sequence genomes, it turns out you don't just sequence somebody's genome brand new. You're always comparing it against the reference, and it has to do with technical considerations. Most people don't realize this. If I sequence your genome, I'm not going to determine your A, C, Gs, and Ts from start to finish, all from scratch. I'm going to actually determine bits of sequence, and I'm going to compare it against this reference sequence, and I look for differences. And that's how these are done. So these 1,000 bases that were mapped, they were mapped because they were mapped as different in a person with some technologies I won't show you. They're mapped as being different from the reference sequence, okay? But remember, in some sense, the reference sequence, it is true, it doesn't exist because it's an ensemble of 50 people. Did I answer your question?
I mean, there are 4 billion genes, right?
There are 4 billion differences at single base. There's only 20,000 genes. Yeah? Okay. Yeah.
So if you got a single nucleotide polymorphism, I could see how that would maybe tweak a protein structure slightly, and you'd have some sort of roughly equivalent protein from one person to another. But if you're sitting here flipping like giant chunks of a gene, I mean, how are you even getting a viable protein out of whatever the gene is coding for?
Yeah, so the question is, he can see how single bases might be little tweaking mutations, but these big structural variants are like sledgehammers, if you will. They should be clobbering genes every time they land in them. And so that is correct, and we've done statistics on this to show that there are less of these on average in a gene than there are in other regions of the genome. So that tells you are selecting against them. If these things do happen in genes, probably very often they are deleterious, and so those, if it happens, that person's not born, and so they're selected against. But I'd be a little bit careful, you know, a single base change inside a coding region can still destroy the function of a protein pretty easily. So it's a bit of tricky business. We happen to be believers that most variation actually affects gene regulation for the reasons you're sort of alluding to. That is, it's a lot easier to tweak regulation and have people live, actually, than it is to start tweaking the genes themselves, because that can kill the gene and then therefore kill the person. Again, not in the person itself, but in their next generation, typically.
So is this just maybe moving genes around that would affect?
Well, I'll show you a few. We'll move to this one, and I'll show you an example in a minute. So the question is, what's that doing to the genes? So these things are all over the genome. There are hotspots, and some of these hotspots do map to disease regions. And some of these structural variants have been mapped to things like psoriasis and Crohn's disease and things like that. So there have been some of these affiliated with diseases. But because they've been relatively new in their discovery, we don't know fully all the ways in which they affect human differences and disease. So the way a geneticist, by the way, looks at disease, if you think about it, it's an extreme end of differences between people. We're all different, but diseases are, of course, all the way at one end of the spectrum there, where often harmful situations come. So a number of them do affect genes. As I say, it turns out it's 17%. And sometimes they'll just lop off a chunk of a gene or maybe lop off the regulatory region or flip it around. And we have some new data that actually says they, in fact, can do just that. But sometimes they'll actually affect the genes themselves. And this is a very interesting example that I'd show to you. This is a case of one of these structural variations that affects olfactory receptor genes. So we have a remarkable ability to be able to smell an amazing number of different odorants in nature, as do most mammals and other, well, virtually all organisms, all animals. And this is one of these examples here. We found a case when we were studying these in which there are two olfactory receptor genes. We don't know exactly what they do. They lie near one another. And it turns out in some people, this region in pink is deleted, and these two genes are fused into one. That's a structural variation. A big chunk of DNA got deleted. So there are people running around. I guarantee they're sitting in this room. There are people who have two of these genes, just like the bottom part shows here. And then there are other people who are fused into one. So we actually followed this up by looking at all of the olfactory receptor genes. There are 851 of these. Some of them are thought to be non-functioning, and that's a whole separate topic. And we looked amongst 25 people. We compared them all to this last individual, and we asked, these are the genes, every one of these little bars is a different gene, and we asked whether there's an extra copy, a loss of a copy, or no difference across all these 25 people here. And the genes are arranged according to where they sit on a chromosome, from chromosome 1 down to there's chromosome 22, and then the sex chromosomes, the X and the Y. And we looked across these individuals, and what you're supposed to appreciate, there are no two identical people in this panel. And I suspect that that's true sitting in this audience, that maybe there are a few, but you get the idea. We're all different in olfactory receptor genes, and so next time you're at dinner and your person next to you doesn't like the main course, well, there may be a genetic basis for that. Maybe he didn't smell it, and whereas you think it's terrific or something. So I do think this is and it's very clear, there's studies from a lot of people that people very much detect odorants very differently from one another. So we think there's likely a very strong genetic difference for this, a genetic basis for this. Yes?
So what's the timing in developing for one of these structural variations to have an effect?
These structural variants, as far as we know, so the question is, what's the timing for these structural variants to have an effect? So if the question is when are these changes occurring, these are mostly germline changes, we think. That is, so they're occurring in our germline and passing them on to progeny. There's an open question how often this happens, what's called somatically, in our tissues as we grow and develop. And it does happen, but it's much rarer. So the differences between two individuals will be much, much greater than the difference between, say, your skin cell and your liver cell. There will be some differences, we think. We're trying to actually figure out exactly how many now. It's a great question, something we're working on. And we think they're there, but they're not as common. So these are mostly germline events. Okay. Yes?
What is gained and what is lost in these?
What is gained means it's gained relative to this individual number 25. Okay, so individual number 25, the way we did this experiment is the reference in this particular. And so we see, so there's a gene, for example, up here, where there's an extra copy in individual number 1, and in individual number 5, there's one less copy of that.
Relative to number 25.
Relative to number 25. Yep. Okay. So I told you the original genome cost almost somewhere from $500 to $1 billion a sequence. We had machines that would basically process 384 samples at once. They were the automated sequencers of the time, and this wasn't so long ago. Remember, this finished through 2003. Well, what's happened is very insightful. there's a major funder of health research. I'm sure you've heard of it, called the NIH, National Institutes of Health. And there's a group in there that actually put out a challenge that said, we want to sequence a genome, a human genome, for $1,000. And a group of companies stepped up to the challenge. Nobody's hit that yet, so don't get me wrong. But what's happened is some remarkably inventive technologies have popped up that are actually starting to push this. And we're now talking about sequencing things for much, much less, over 2 logs less than they were just a few years ago. And these are just some of the companies that are out there, and there's a whole series more in the pipeline that are going to even leapfrog the ones I'm showing you here. But this is the first of them, 454. Then there's some of these called Illumina, Life Technologies, Helicos, and here's the latest one that hit the scene. And they all make claims, and it's hard for me to tell exactly what the real numbers are. But I'll show you some numbers in a minute. I mean, we can get a pretty good idea what the ballparks are. So what's different about these, this one makes now about 450 nucleotide long sequence reads. These, amazingly enough, are very short. When they first came out, Illumina and Life Technologies, they were only making 36 or so bases. Now they make 50. Illumina's making somewhat longer. But these are relatively short reads. They're only 100 bases, right? And you've got to get 3 billion bases deciphered. So how do you do it? Well, the way you do it is the way I mentioned before. You get, what's remarkable about these technologies, they make very short reads, but they make millions and millions and millions of them. So a single run will, in fact, make somewhere on the order of 160 million, say 50 base or 100 base reads. It depends on the instrument and the technology. But you can get very, very large numbers. And even though they're short, and they often have high errors, but you can get so many of them that you can find these single nucleotide differences pretty well. And the way you do that is, again, you don't try and sequence a genome de novo, you match it to the reference. And so when you get your sequence determined at the end of the day, you're not really getting your sequence determined. You're finding the differences between you and what they can match in this reference genome. And that works really, really well for these single nucleotide polymorphisms. It's a lot harder. You can get some of it for these structural variants, but you can't capture all of it. There's no good technology yet for capturing all of that, but you can capture a certain amount. So we still have a ways to go to really be able to properly sequence human genomes. And again, there's some very interesting technologies that are coming in the pipeline that will make things more and more accurate. But nonetheless, we can get a pretty good draft of a person's sequence. And then there's a big argument, well, gee, does that count as a sequence or doesn't that count? I don't want to go there. But the bottom line is we can get a lot of information, genetic information from a person, even if it's not perfect. Yes?
So who are those 50 people? That doesn't seem like a very big sample to me.
Well, it's even worse than that because it got, and if you want the complete story, there's a few people who dominated the pool. And they happen to be, so their sequence is overrepresented relative to others. And as it turns out, it's Caucasians. So the reference genome has a lot more Caucasian information than that of other ethnic groups or geographical groups. But if you want to get even the more complete story, there's a big race on when this was all happening. You may be familiar with this. There was a public group that ultimately made the reference genome that everybody's using. But there was a private group led by a guy named Craig Venter, who marched ahead and was doing this separately. And in the end, he sequenced himself. So the private group was Craig's group sequencing Craig, and the public group sequencing this pool. So, and that actually then was, in fact, the first true individual that was the first personal genome that was sequenced was Craig Venter's. Okay, was there another question down? Oh.
Actually, I have a question. I seem to ask it first, but is there any work being done to augment the reference genome?
So the question is there any work being done to augment the reference genome? And the answer is yes in several ways. First of all, even when it was called done, it wasn't really done, and it's still not done. There's about 300, I believe, gaps, maybe 200 or so. I don't know the final number right now, but so there still are a few holes there. And the other is, it's not done because it's not a real sequence. If you start comparing with other people, I'll get to this in a minute, but there are other variations out there. So there are projects to capture all the variation out there. And it's also not done because we don't understand all the information that's in the genome, the regulatory information we talked about earlier. And again, exactly where all the genes are and what all the genes are and certainly what they're doing, we're a long ways from done there. So there's much to be done. Okay. Okay, so these new technologies that have come along have, as I say, dramatically dropped the price of sequencing genomes. And in fact, this red line then is the cost of sequencing as a function of time on the x-axis. And you can see it's kind of marched along pretty good until these new technologies have emerged. Now, I imagine many of you have heard of Moore's Law, right? Yeah. Does anyone, so who can tell me what Moore's Law is?
Every 18 months, computing power doubles.
Yeah, so capacity doubles every 18 months. So that certainly wasn't happening with sequencing, but if you look what's happened with these new technologies, they're actually increasing about 10-fold every 18 months. It's much, much more dramatic than Moore's Law. Okay, so it's really going incredibly well. What's happened then is the cost of computing, storing the information, and actually analyzing the data, it's expected will be higher than that of actually sequencing the DNA pretty soon, if not already. So so you can see the challenges are shifting from what they used to be. And with the technologies now, it's hard to get a firm estimate on this because everybody compares things a little bit differently, but it's roughly on the order of about $50,000 to get your genome sequenced with the limitations I just mentioned. You get lots of these short reads, you can figure out most of the SNPs and a lot of these structural variants, but you won't figure them all out, but you'll get a lot of information, okay? And that would be a personal genome. So there are projects, as I just mentioned a minute ago, to actually now expand on this information. So there's a project that's been launched to try and understand human variation across the globe. And this is called the 1,000 Genome Project. So it's a consortium involving many hundreds of people to sequence 1,000 genomes using these new technologies and try and understand, or at least capture, all the variation that's out there. And this is not really to necessarily understand disease just yet. It's just to try and understand variation that exists around the globe. As I said before, there's a big push on to sequence a human genome for $1,000. Now, I said not so long ago I'd probably get my genome sequenced when the price hit $10,000. But the way things are moving so quickly, I think I'll wait a few years, and I think it won't take long before it's only a few $1,000s, I'm sure. So, but this is something you can think about, and we'll get to this in a bit. So with this then, now it's the blue line that's showing the cost in sequencing. Sorry for the color change. With this dramatic drop in sequencing, people's genomes are just starting to get sequenced. So that's supposed to be 0. First individual genome sequence, not the reference sequence around here, and so on and so forth. So now there's 8 genomes that have been sequenced, and I think everybody's expecting this to skyrocket. And on top of this, there's a number of disease genomes that are getting sequenced. There's a big project out there to sequence cancer genomes and sequence large numbers of these with the goal of trying to understand the genetic bases of lots of different kinds of cancers that are out there. That's another large consortium project that exists, and some of these first cancer genomes have just been released. And we don't know for sure the disease-causing genes in those, but there's some very interesting candidates, if you will. And the hope is that they'll lead to a platform for discovering new genes involved in cancer. Yes?
So I'm assuming that the first one over there is a Craig Venter that you mentioned earlier.
Yeah, this must be Craig.
So, so once we get to a few thousand people that are sequenced, will that change the reference genome then to be the conglomeration of those few thousand where
yes.
we don't really know what your genome should look like?
So the question is, once we get to a few thousand, will that change the reference genome? And not really, but it does raise the issue. You're hitting on a really, really good point. What should be the reference genome? Because right now it is a conglomerate. It's a one-copy genome too. It's not even 2 copies, right? And there's no really good answer to this. What should be used as the reference genome? And I've heard interesting proposals out there, and it just hasn't been resolved. And at this point, the reference genome may just be the one we have, even if it's imperfect. and it's, in a sense, it's a virtual genome because it is a conglomerate of individual sequences. So I think because of the way everybody's mapping things, but I think the main point is that we make sure we capture all the variation that's out there so we understand what each person's sequence is at each position. Because at the end of the day, what I would like to see us do, and I think this is my, these are the three, well, I'll get that in a minute. We're going to have many, many sequences out there. And I think this will be a very, very powerful data set if we know the information associated with each individual along with the DNA sequence. Now, there are other issues that are involved with what I just said as well that we'll probably touch on later in the talk. Yes?
The sequence is as tedious and expensive. When we read of criminal activities where somebody's accused of a rape, for instance, and they check the DNA and compare it with some residual on the victim, what are they really checking? They aren't doing the if it's not the sequence.
Yeah, so he's asking about criminal events when during a rape, for example, what are they actually checking when they check DNA on a victim? And the answer is they're actually checking markers, specific markers in the DNA. So regions of DNA that do vary between people, they're checking that. And they check enough markers that it's like a fingerprint, if you will. Something that's characteristic that by probability can only be associated with a particular individual and not other people. So if you check enough markers.
Is that part of the nucleotides in the structure when you go through the...
It is, yeah. So at the end of the day, those would, so the question is that part of the nucleotide structure? And the answer is yes, it's part of these nucleotide differences that I mentioned earlier. It's not the whole genome sequence, it's just little snippets, but enough to tell you that one person's different from another. Yes?
As one sequences the genome and you define these differences, couldn't you look to define statistics around each of the differences and ultimately look at those statistics to come up with a more probable reference genome?
Yeah, so the question is, can you use statistics to knowing what's going on at each position and use that somehow to call a reference genome? I mean, I think what you're talking about then is a consensus genome. That is trying to say, since most people have an A at this position, let's call that an A. And since most people have this, but to be honest, that's also a virtual genome as well because nobody's going to have the consensus at every one of these positions. And then also, I'm not sure what that means because, again, it's going to be based on whatever population you're sequencing as well. So if we sequence a bunch of Europeans, they're all going to have a certain sequence, and that'll be different from a bunch of people from Yoruba, for example, or from China, who will have a different one. And I guess if you want to do it straight on numbers, maybe you would say the Chinese, I suppose. Anyway, yeah. So you see the issue. It's not that. At the end, I think the main thing is just to have something that you can compare against. And what's most important is the functional information that's deduced from these sequences, which is not that easy, by the way. We'll get to that in a minute. So anyway, these are some of the genomes that first got sequenced. We have Jim Watson, Craig Venter, and we even have one at Stanford. Steve Quake just sequenced his own genome with one of the technologies that I mentioned out there. So you probably may have seen some press releases on that. So these are 3 of the 8 individuals that are there. So I'm a believer that there will be, in fact, 1 billion human genome sequences done. And this is something for you to think about. Virtually everyone in this room will have the opportunity to get their genome sequence if they want. Now, you may not want, that's a separate issue we'll get into in a minute, but I think cost-wise, this will be affordable for most people. And then you might ask, well, why should we be sequencing genomes? Maybe I should have led off with this. And there's a lot of reasons for wanting to sequence genomes. One is getting back to that fundamental question. We'd like to understand how a genome comes to life. How do you get from DNA to a person? What are all the parts? How do they work together to be able to build a living organism? And why is it our genome builds a human being and a chimpanzee builds a chimpanzee genome? How is it? They're very, very similar to one another, yet the organisms at some level are fairly different. And so I mentioned before, we'd like to understand why we're different from one another. And I mentioned the species one just now. And for this talk, a big issue is we'd like to understand the basis of disease and try and use information from the genome to try and improve human health in various ways. There are through diagnostics and therapeutics, and I'll touch on some of these in the remaining time here. One thing I can definitely say is that genome has had a huge impact on discovering genes that cause disease. As I said earlier, there's been a handful of genes that were identified that were responsible for causing human disease. You've heard of them: sickle cell anemia, cystic fibrosis, fairly common diseases. But after the genome got sequenced, and after we found a number of markers, these single nucleotide polymorphism types of markers, it was suddenly possible to map lots of different diseases. And in fact, there's now something on the order of over 200 disease genes that have been identified just in the last few years. And this number keeps skyrocketing because it's now the technology is so much simpler based on the information that's come out of this. So this has been really remarkable for helping us understand and identify disease genes. Doesn't necessarily cure help us for a cure for those diseases, but it certainly helps us at least understand the underlying genetic basis, and then gives us angles, we hope, to be able, at least at some point, cure disease or at least diagnose disease. So let's touch on this issue. Do you want to get your genome sequenced? Here's why you might. You might want to because you might ultimately want to know your sensitivity to medications. We'll go through some of this in a minute. You might want to know if you're likely to get certain diseases. But the number one reason, if you ask people who have gotten themselves either genotyped, meaning had markers with one of these services that you've seen the balloon flying around on, say 23andMe or Navigenic, most people do it because they're just curious. They want to see what's in their genome. And if you get your genome sequenced, you'll get even a lot more information. Why you might not? Well, you might not want to know if you're likely to get certain diseases, and there are many incurable diseases that are out there that you can diagnose and do absolutely nothing about. Huntington's disease is a good example. And some people would not like to have that information, and other people would. And there's no right or wrong to this, it's just the way it is. But it's up to you to decide if you're interested. This one I'm going to touch on more in a minute. Most diseases are complex. So even if you get your genome sequenced, you might think you're going to understand a lot when you get your genome sequenced, but the answer is you probably won't, because we only know the genetic basis of, say, 200 or so genes that you can actually do anything about or even understand. And most of those are incredibly rare, so the odds of you having those are pretty low. There are a few common ones that are relevant, but there's not enough information, I'll get back to this a little bit later, to know how to interpret a lot of our genome information. I don't think that will be true forever, but it's true right now. And lastly, I'll also touch on this issue. You worry about genetic discrimination from various entities, and we'll touch on that a little bit in a minute.
So we go to a company like 23andMe or one of the other ones, and they do the sequencing. Are they only doing the 200 genes that we know of, or are they doing that much, or how much are they actually doing?
Those companies, so the question is, what about 23andMe and other companies? Are they doing a limited amount of sequencing? No, they're doing something different. They're doing something called genotyping. They're basically identifying genetic markers. So instead of sequencing the whole genome, they'll identify, for 23andMe's case, they'll identify 600,000 markers, which are these single base differences throughout the human genome. And based on these markers, they can say if sometimes these markers are linked to diseases like diabetes and things like that. So they make estimates based on that, of what the probability is. I think they're fairly careful about saying that these are not, you know, these are, you might have a twofold elevation for something, for some particular disease over something else, but they'll certainly, that doesn't mean you're going to get this disease. A twofold, you know, maybe instead of being 1 in 10,000, now you're 1 in 5,000 chance or something like that. You see, and none of the data, or I shouldn't say, there's some of these markers that are fairly predictive, but the bulk of them are not, you know, not strongly predictive, shall we say. So the information is somewhat limited, although there is some interesting information. But in the long run, this information will be very valuable. But right now, it's fairly limited. Yes?
How do you tell where you come from?
The question is, how do you tell where you come from? It's because they're markers, which you can get either using these genetic tests or from your genome sequence, that different ethnic groups share. And it's pretty amazing. There's somebody who's going to be joining the Stanford faculty soon who actually looked at this in detail by looking very, very carefully around the world, not through genome sequencing, but doing fairly high density markers. You can actually pinpoint to where your family comes from within about, I think it's 170 kilometers, 170 miles, something like that, within Europe. And it raises this issue about when you get your genome sequence, are you going to somehow be identifiable? Okay, because they'll certainly know where your family comes from. Now, obviously, people move around, so it's not exactly where you're living this minute, but it's where your family, has been living. Your larger family has been living for some time. So your ethnic groups have certain markers. Some markers are fairly recent, and some are very old, and that's been very, very valuable for being able to trace human evolution and human migration patterns as we've moved out of Africa and populated the rest of the world.
The genome sequencing pick up all the mutations like the BRCA mutations?
Yeah, so does the genome sequencing pick up BRCA mutations and things like that? And the answer is absolutely yes. It also picks up, well, if you sequence your genome, you will identify all that information. And that I would call as incredibly valuable information you want to have. Because if you do have mutations in those genes, you'll get yourself checked regularly, or you should. So that's something you do something about, which is a good thing. There are other cases, for example, ApoE, which is implicated in Alzheimer's. Some people don't want to know. So Jim Watson, when he got his genome sequence, said, you can publish my sequence, but not my ApoE, because he doesn't want people to know if he's going to get Alzheimer's. Now he's like 85 years old or something, so I think we probably know the situation already. And as far as I can tell, he's still quite bright. So, yes?
Is there some evidence that we had a very limited number of ancestors at one time that we evolved from?
Yeah, the question is there some evidence we've had a limited number of ancestors? And yeah, humans have gone through bottlenecks in our evolution. There's some incredibly good groups at Stanford, Marcus Feldman, who's looked at human migration patterns and these sorts of things, and he would certainly be a good person to talk to about this, but the answer is yes. And, yeah?
Discrimination issues, these companies that would go against more consumer-based typing or comparison.
So
What are the current laws about releasing this information, and who really does have access to it?
Yeah, so the question is discrimination, and I'm going to touch on some aspect of that. On the issue about releasing information, this is a very serious issue that is out there. So there's been a lot of different ways to approach this. Some groups that have been involved in human genome sequencing, like George Church's, only takes volunteers who are willing to put their DNA sequence out in public, meaning they'll get it sequenced and then they'll put it in the public domain and they're not worried about it. Okay, and he's got I've forgotten what the latest number is, but he's got at least over 1,000 people signed up to do this. So then there's no issue for those individuals. For others, this could be an issue about consent issues and getting yourself, if your DNA or your cells are used, it's believed you should get proper consent. And if you're giving up your samples for something and you don't want your genome sequenced out there, you might make sure you see if you're giving up these samples about what's going to happen to them if you don't want your sequence out there. These are issues because you ultimately, if they took one of your samples and sequenced, it's possible you could get identified from that. And that may not bother you, but it may bother you, and you don't want other people to know. So these are issues. There's other very interesting issues too, because if you decide I'll get my sequence, my genome sequence, well, you just released half the sequence of your siblings without their permission, perhaps. So there are issues like that, or your parents get themselves sequenced, or your parents got sequenced and you didn't give your permission. Well, if your parents get sequenced, if both parents got sequenced, well, your sequence is in there somewhere. You don't know the exact combination, but it is in there. So you see the issues about this. And My own view is that I hope we can get to a culture where this is not an issue, that people aren't afraid to get their genome sequenced, and that we can share this. That's a very utopian sort of view. I can imagine not everyone shares that. We'll talk a little bit more about this as we go along. All right. So I wanted to spend some time, we're in great shape, talking about the impact of genomics on medicine. And this is really, I want to emphasize, in its incredibly nascent stage. This is incredibly early days, and there's much to learn and do ahead. But I'll tell you where we are now, which is not very far, but we're trying to give you a sense of where we might be as we go forward. So the biggest impacts on genomics are the first two listed here. Predisposition testing. What are you potentially at risk for? Cancer is certainly one of the most obvious. And the other is something called pharmacogenomics, using your genetic information to help derive treatment or predict outcomes and that sort of thing. And lastly, there's this issue that we just raised about how one might use personal genetic information. Certainly the biggest impact in genetic testing has been BRCA1, BRCA2, which are fairly common mutations in women. And you almost automatically know there's something there because there's a family history of breast cancers, how it's first found, but now it's very clear BRCA, at least BRCA1, I believe BRCA2, have been implicated in ovarian cancer as well. So if you have these lesions, if it's running in your family, it's almost always recommended you get tested for these. And as you should, because what will happen is if you are tested and you are found to be carrying these mutations, you need to get yourself monitored very, very frequently so that you can catch things. Cancer, especially for solid cancers, if you catch it early, it's virtually always curable. Not always, nearly always. But if you catch it late, it's almost never curable. So the key is catch it early. And so if it's clear you have a genetic disposition for this and you're carrying these mutations, you should get yourself tested quite a bit so you can catch it early. And for some kinds of cancers, breast cancer, it's a lot easier to catch earlier. Ovarian cancer, as you might imagine, is really hard because it's not like you're going around getting your ovaries tested all the time. So, but if you know you're a BRCA mutant, you will get scanned and more often than not, and therefore can catch things much earlier than you would if you did not know this. So this is a clear case where there's a genetic test and it does have an actionable outcome. That is, you should be behaving differently than if you did not get that genetic test. And if you don't carry the mutation, then you don't have to worry. You should probably still get tested periodically, but not at the same frequency. Okay? So that's a very clear case. There are others that are interesting. These are not very common. This one I think is quite interesting. There's something called long QT syndrome, which is a series of series of different genes that are out there that affects your heart rate and such. And individuals with this can have arrhythmia issues. And one particular type of this disease is actually if you get startled in your sleep, these individuals will go into cardiac arrest. So obviously, if you're carrying this, you shouldn't startle those people. So, and also there are drugs that can actually work on these individuals and stuff too. So this is a very, very rare disease. I don't know if, I won't ask anyone to disclose what they have, but it's unlikely, but it's not a common disease. And then there are others coming along, such as type 2 diabetes and such, where there have been genetic tests as well. So these are usually diabetes is almost always discovered through glucose levels and stuff first, but you can subtype these, at least in this particular case. There's one here. And there's getting to be more and more of these. Most of these are rare. Again, the BRCAs are the most common. There's a new one coming out for breast cancer that's not quite as frequent as the BRCA mutations, but has just been implicated as well. So you'll see more of these as we go along. And the expectation is, again, this list will get longer and longer. So that's predisposition testing. Are you likely to get a disease? Other things you could get tested for are Huntington's, Alzheimer's. There's a number of Parkinson's. There are certain genetic diseases there, and some people, again, choose to get tested for those. And some of these are incredibly strongly predictive, meaning if you are positive, there's very, very high probability you will get the disease, and others are less so. So, but anyway, these are, and again, some people choose to do this. There's nothing right now you can do about those. So other people don't want to get tested.
Have they determined that there is a genetic predisposition for Alzheimer's?
Yeah, so the question is there a genetic predisposition for Alzheimer's? So at least for one test, the answer is yes. These are often not single genes that are involved in these sorts of things. That is, so for some individuals, yes, you can be fairly strongly predictive of Alzheimer's with certain ApoE alleles. ApoE, I should say, mutations, or they're not always mutations, they're called them variants.
I believe there was a time not long ago when the medical profession believed you couldn't ascertain if somebody had died of Alzheimer's without autopsy, right?
Yeah. So the point was raised, you couldn't tell if somebody died of Alzheimer's unless you had an autopsy. And by the way, so not everybody who gets Alzheimer's has these ApoE. So there are many other types of Alzheimer's that can come along with this, just like not all breast cancers involve BRCA1 or BRCA2, if you follow. So there are some genes that are strongly predictive, but there's lots of other things that can contribute to these as well, and Alzheimer's is one of those. So I've forgotten what fraction of the population is assignable to ApoE, but it's not the majority, I don't think. I'd have to look that up, so you can email me if you like. Okay, pharmacogenomics. So this slide is really meant to lay the landscape for the issue, and then I'll talk about some specific examples in the next one. So as you probably appreciate, and I'm sure you've seen this either for yourself or family members, people often take medication and nothing happens. And these are just some numbers here for some very, very common situations: hypertension, depression, diabetes. For each of these are large numbers of people we're talking about. You can see there's a significant fraction of people with any of these three diseases who do not respond to treatment. Okay? We don't know why they respond to treatment. There can be lots of different reasons. One is maybe they're not metabolizing the drugs the same as other people, and so on and so forth. And we'd like to be able to understand those who are going to respond, if you think about it, to a treatment from those who don't. You'd save a lot of time, a lot of money, in certain cases, many lives, if you knew who was going to respond to a drug and who wasn't. So this has ultimately got very, very important for the genetics and human health. Okay, and so these are some of the factors I've written at the bottom here in which genetics can influence treatments, drug concentration, and whether a drug is active against a particular target. I'll explain this in these next slides. So there are really two ways. One is dosing drugs, and the other is will a drug be effective against a target. They're not exactly the same. And that's explainable here. So we have proteins in our body that are encoded by particular genes called the cytochrome P450s. These are actually very important molecules for us. They make our hormones. They detoxify things in the environment, and we use them for all kinds of different things. And they are involved in drug metabolism. There are many genes. There are 80 of these cytochrome P450s, and actually they show differences in expression between men and women. So if you and your mate, if a man and a woman don't respond the same, it's because they may have real physiological differences. These are not expressed the same in men and women. So there's now several cases. The most famous is this one, warfarin. It's an anticoagulant used quite a bit. And this is one of these cytochrome P450 genes, the CYP2C9. Sorry, genetics has weird names. I've tried to avoid that in this talk, but this one does exist. This is not a cytochrome P450, this VKORC1. It's a different gene. It's a reductase that also modifies compounds. And basically what these do, there's what are called two different alleles, meaning two different versions running around in the human population. There's more than two. But for each of these genes, there's at least two different versions around. One version makes people metabolize this drug a lot faster than the other version. So if you metabolize this drug faster, that means you need more of it for it to be effective. So what they now have, there's a commercial test that will test which of these variants you have, and they know how to better dose you for the drug based on that test. Does that make sense? Now, this is one example. We don't know what most of these 80 do. So you can see that ultimately we'd like to match every single drug up with every single metabolizing enzyme, and then in an ideal world be able to match that. That hasn't been done, but that would be an ultimate goal of genetics, to be able to match drug doses with the particular variants of these different alleles, of these different genes that are out there. So that's a drug dosing example. There's another one, not as common, that exists as well. How about targeting specific diseases? Well, breast cancer, you hear this a lot for cancer. People throw out names: breast cancer, colon cancer, leukemia. These are rarely single diseases. They're very heterogeneous. There'll be several types. They all look like breast cancer, but this is a classic case. Breast cancer can have something that are called HER2 positive, meaning they're expressing this protein. It's a growth receptor called HER2. And it depends on the population, but somewhere on the order of about a quarter of people with breast cancer are HER2 positive. Okay, if you're HER2 positive, the best therapy is an antibody against HER2. Okay, makes sense. But of course, the other 75% aren't HER2 positive. It would be worthless to throw HER2 antibodies against those people. It's very expensive, first of all. But second, it would have little or no effect. And so therefore, you match the drug to the particular disease, the exact disease you have. Now, there aren't huge numbers of examples of these, although, quite frankly, there are plenty of breast cancer patients around. So actually, it does affect many individuals in this particular case. But there aren't many different kinds of examples, I should say, out there. But it's the early days, and so the expectation is that we will get better and better at matching these drugs. Here's another amazing one that I have a personal experience with, this one, Iressa. It's used to treat non-small cell lung carcinoma. And only a small fraction respond, 10%. But that small fraction turns out just about get cured. It's amazing. Their tumor just regresses completely. Ultimately, they may develop resistance, but at least for several years. I should say this is usually diagnosed late. So when people get this, they usually have several months to live. So my cousin actually got this when he was 42 or so. And so fairly young, and it was diagnosed late, and they had a certain window in which to treat him. Now, unfortunately, he was a non-responder, so they did go through an Iressa treatment. He did not respond, and he ultimately died. But during that window, you can see, now they know, we didn't know at the time why it worked. It's now clear that it's very specific mutations. This is a kind of diagram of a protein. This won't mean much to probably 95% of the people here, I suppose. But this is a diagram of a protein. But the drug only binds to this protein if you have a certain mutation, okay? And a certain kind of mutation. So only the few people who have those mutations, these 10%, will respond to the drug, and they respond in an amazing way. It binds this compound, and it basically affects this whole pathway this protein works in and cures the individual. But the other 90% don't bind the thing, so it's completely worthless. And so now there's a genetic test. If you have this, they will test you to see whether you have one of these mutations. If you do, you take the drug. If you don't, you do something else. So you can see the power of matching drugs to the disease. Why waste time on something you shouldn't be doing? And again, this is a matter of life or death. This is not something you want to mess with. This is, you want to do it, you want to do it right. And so there aren't very many examples, but this is what geneticists hope is the future. We want to be able to match drugs to the exact disease and be as effective as possible. Yes?
When you say mutations, do you mean structural variations or SNPs?
In this case, so the disease, I didn't get into this, but the party line is that many forms of cancer, most cancers arrive by, say, 5 mutations or so. Usually, they can be structural rearrangements, they can be point mutations. In this particular case, they're point mutations. I forgot to repeat the question. So the question is, what do you mean by these mutations? And what I'm saying is these particular ones are point mutations But cancer can arise by any of these things. They can arise by structural types of variations, and they can arise by point mutation. And every cancer usually has multiple mutations. Not always, but usually. Well, you guys ask great questions. All right, so you're not really supposed to read this, but the point is, you know, it looks better here than on my screen. All right, so the point is there's now getting to be a lot of different genes for which either some sort of response has been measured. It's still a fairly small list if you think about it. We have 20,000 genes, and the list at most, it's not even 200, or maybe it's 200, but it's not huge. And most of these are often rare situations. But we are starting to be able to match drugs and treatments with diseases. But again, it's incredibly limited so far. And I've shown you the best examples that are out there, the HER2 and the RS, and there's a few others like that. Okay, there was an interesting case. Yeah, I think we probably have time for this. But, you know, one issue that's come up is, can you match drugs to ethnic groups? And this was controversial at the time it came out in 2005. There's a particular, called a combination therapy, which means you add two things together to get an effect. And it's called BiDil. And it's this combination of two things. First, it was tried in a general population. It wasn't found to be successful. So then they tried it in an African-American population and did seem to have a positive response when compared to a placebo, as said here. So it has been used for a while. It hasn't been a remarkably successful drug. It also goes to show how you deal with this. The biggest issue is that African Americans are an incredibly heterogeneous population. And anyone, the way this is used, it's all by self-reporting, meaning a person self-identifies himself as an African American or not. And since African Americans are quite mixed often, it's not the best way to actually be doing this. And so it hasn't turned out to be quite as effective. So it may not be the best way. It shows how you probably shouldn't stratify a population. You should really probably have good genetic tests to see if you could really match this stuff up. That was the lesson I guess I was trying to say. So we'll see what happens. That drug isn't being used a whole lot. So just to recap, what are the advantages of pharmacogenomics? Well, as I've implied already, it's targeted therapies. You only want to give drugs to patients who will respond. I didn't talk about this, but in an ideal world, you want to give drugs to patients who will not have adverse side effects. I'm sure you're probably aware that there are a lot of drugs out there that are incredibly successful at some level, but are problems for a select number of individuals. And certainly Vioxx is a classic case, right? Vioxx has been associated with heart valve defects. And there's no question there is that correlation. But Vioxx actually helps a lot of people. So in an ideal world, you'd like to be able to stratify away those who have the adverse side effects from those who don't. I'm not saying you can do that with Vioxx. Maybe you can, maybe you can't. But drugs get killed because they will have an adverse side effect on a small population, and yet they can be incredibly beneficial to others. And so there are a lot of people who are upset about certain drugs when they're pulled from the market because it's the only thing they'll respond to. So you hear about all the negatives, but there are positives that can be an issue. I mentioned the issue about, well, I guess I just touched on item 3. You could therefore rescue drugs that may fail if you can figure out why they're failing on a set of individuals. And I meant number 2 is a matter of giving the right doses. We talked about that already. What are the problems with pharmacogenomics? Well, probably the number one is economics. If you think about it, a drug company doesn't want to stratify its market. It doesn't want to make a diagnostic test to go along with every drug. It's expensive. It doesn't always work. It's a pain in the butt. So what do you do about that? Well, right now, nothing. I mean, it's better for them to take it's a lot easier for them, I should say, to make a drug that everyone would take than to try and stratify the population. I think as the competition gets higher, though, in certain areas, I think you probably will have to stratify the population. The second thing is that sometimes these studies, if you think about it, they're typically done in one particular population and may not be generalizable across all populations. That is, a trial may be done in one area where there's one ethnic group that predominates, For example, maybe it works in people of European descent, but it may or may not work in those of Asian descent or in African descent and such. And the trials may not have been geared to actually see that. They may be heading towards one population or another. So these are things that you have to look at. And probably one of the most important things is something I touched on earlier, I want to spend a few minutes on now, is that most effects we think are out there are not due to, most genetic effects are not single gene loci. So the cystic fibrosis you've heard about, which runs very purely in family, is a single cell locus, sickle cell anemia. But there's a lot of drugs out there that are not easy. You can't assign a single gene to them very easily. I'll go through this in a minute. Most diseases, so I guess to recap, few loci are strictly predictive of disease. Where you have this, you're going to get the disease. When they're there, you see them, again, but they're not that common. Most common diseases, diabetes, which is on the rise, schizophrenia, autism, even asthma, which has probably not been studied as much. These are very, very common diseases, right, in the population, amazingly common. It's very, very hard to find the disease-causing genes. And let me give you one example. Diabetes, they just did a recent study where they looked at 30,000 disease cases and 30,000 non-diabetics and looked for the markers to map, I won't get into details, but to map all the genetic diseases responsible. We know there is genetics causing diabetes. So they looked at this huge number of people, tried to map all the genes. They found 18 genes involved in diabetes. You add them all up and you can account for 7% of the genetic contribution of diabetes. So a huge study can only find a tiny little bit, and they're spread over 18 different genes. So what that says is there's not just one dominant gene out there that's causing diabetes. It tells you there's a combination of effects. We don't fully understand this, but there's probably two major likely scenarios. One is it's a combination of different genes. So you get the wrong combination, you get diabetes. And the other is that, they call that gene interaction. And the other is that you might have genes in certain environmental situations. So the genes plus a particular environmental situation leads to diabetes. Those are the two most popular thoughts for diabetes. And this is true of other things. The easiest one to explain to you, though, is this one I listed up here about height. The best way, height we all know is genetics, right? You all see your parents and such. Everybody knows the Belgians are the tallest and the Southern Europeans are shorter if you look at a European map. Very, very obvious. The best way to tell height is to look at parents. People have gone in and looked at large numbers of individuals to map the genetics. And again, a few loci have been found, and they account for this tiny little bit of the genetic contribution of height. We can't explain height by genetics well. And so looking at a person's parents is the best way to tell height, okay, in spite of all the genetics and such thing. I hope that won't be true in the long run. That is to say, I hope as we understand things better, we'll say, oh, there's 30 different genes involved in height, and you have this particular combination. And all you who raise your hands about your computational skills, we need you for these kinds of problems. We see the right combination leads to whether you're tall or short. Okay, yes?
When they look at like that diabetes study, you said that they looked for genes that cause diabetes. Do they also look for variations that prevent diabetes?
I don't know the answer to that. My guess is no, because it's hard to do that in the way the study was structured. I think the answer would be no. You'd have to somehow know that there are sets of individuals that don't get diabetes and it's somehow running in that family. It's not, because it's not that common, it's not that easy to do that kind of study. There are diabetes, so as far as I know, nobody's done anything like that. There are other diseases that has been done for, where you look for people who are protected, including for heart disease, for example. There are certain populations that seem to be resistant for heart disease. Amazing, right? So I forgot to repeat the question, but sorry.
And I assume in your discussion of diabetes, you were talking about type 2, not insulin-dependent.
Yes, yes, yeah, thank you, yeah. Okay. So the question was type 2, I was talking about type 2 diabetes, the answer is yes. Okay, sure.
You haven't said anything about genetic predisposition toward addictions to chemicals or other things. Is there such?
So I haven't said anything about genetic predisposition to chemicals and addiction, and is there such? And the answer is yes. There have been genetics associated with all kinds of things like addiction, dyslexia, so various learning things. It's amazing what's out there, and they're very, very hard things to map. A gene has been linked to dyslexia, by the way, and I know people are working on addiction. I haven't heard of one yet. Yes?
Is it possible that this kind of genetic view of disease or predisposition is actually only a very minor part of it, that what's actually happening here is a systemic interaction with the environment, chemicals, and that it's almost a random situation in terms of creation of the disease? You mentioned a number of those diseases, but in those cases you only talked about a very small percent identifiable with very specific regions or combinations of genes. And you didn't talk about the effect of
So the question is it possible that's really the system, such as the environment and things like that are contributing to that? And the answer is yes, but again, remember, there is a genetic basis to these diseases. You can follow diabetes in families. It's not always 100% penetrant, meaning it doesn't happen 100% of the time, perhaps for some kinds of diseases. But you do see it in families. You do see it. It's greater than random. So there is a genetic component. What I'm saying is when they map that genetic component, they can only find some of it. There still is more genetic component to be found. But the point you're raising is exactly right. We think it's not just the genetic component. It's either the interaction of a bunch of genetic components or the environment, as you just pointed out, can still be a strong factor. And there's no question the environment has a very strong effect for many, many types of diseases. So, but there is a genetic component to this. And again, all that's saying is that this isn't simple. That means it's going to be combinations, and it's our mission to figure out these combinations so that we can do something about it, either avoid those combinations or make sure we're prepared to be ready with therapies and things, okay, or careful monitoring of the disease in affected individuals. Okay, we'll start back and then move up.
Yeah, you're looking at the genes for disease components, and you're finding a little correlation in many cases. Since you're not looking at the other 98.5% of DNA, how do you know that there's no contribution there?
Oh yeah, great question. So the question is, because we're only looking at, we're not looking at the other 98.5%, maybe we're missing stuff there. And the answer is we actually are looking at a lot of that 98.5%. That is to say, the markers are all over the genome. So you're still looking for this linkage. And it's getting a little bit technical, but the point is they are actually looked for. So you would find it if it were there. The answer is they're just small contributions. Yeah, I'll leave it at that. So the bottom line is that we're not ignoring the 98.5%. Now there are times when you're trying to zoom in, you find a region that's causing a disease, and you try and zoom in and find the gene responsible. So you find a big region, a big chunk of DNA, and in there's a couple of genes, and you're looking through these genes to try and find the mutation, and often you find it's not in the gene. Then it's often in that, that other non-gene part that you should be looking at. And more people are learning to look that way. So your point is very well taken. Okay. Did you have a question as well?
After this first cell is created by parents and the person takes birth, does this DNA structure change over the period of human's life or for whatever reason, variations or chemicals?
So the question is, after something is found and then a person takes drugs, does this DNA lesion change? And the answer is it can. So in the ERESA case I mentioned, what can happen is individuals start developing resistance to that drug. That is, suddenly a mutation will pop up that will no longer bind that. And then suddenly the tumor will start growing again. So that can happen. And so these resistance things do happen. And unfortunately, a lot of things they use to treat cancers and such are actually mutagens themselves. So you do have to worry. It does make people, so often these are nasty chemicals and they can make people sick depending on the particular cancer and the particular treatment. So that kind of thing can happen. Other complications can arise.
All these radiations, do they also cause mutations which can cause diseases?
Yeah, certainly radiation. The goal is, so the question is radiation, these other things cause cancer and such. So it certainly damages the cells, and the goal is to kill them, but it definitely has got to cause problems as well. Yeah, it's banging on things, with a sledgehammer again. It's not at all targeted therapy like you'd like to have. Yes?
Can things like blood transfusions then bring genetic mutations into someone that didn't have it already and then cause them to get cancer?
Can blood transfusions bring things into people and cause cancer? And the answer is it would most commonly happen by an inadvertent virus being transferred, such as hepatitis or something. I can't think of a situation. I guess theoretically it's possible. I haven't heard of that, but that doesn't mean it doesn't happen as a general. You'd have to have these cells go back and populate the bone marrow or something. I think I'd worry more about a viral infection type of thing rather than some sort of genetic disease transfer.
If you get a genetic mutation from an environmental factor like radiation or something,
right?
Can you then can you pass that on? Would that pass on to your children?
No, these are almost always, so the question is if you get a genetic mutation from an environmental situation, would you pass that on to your kid? And the answer is almost, I would assume nearly in every case no, because these are usually what are called somatic mutations. They're happening in your more adult tissues, and your germline is usually protected from that. Okay, so it would be very rare that would pass into the germline. Okay, yes?
So building on that, there's a lot of discussion about epigenomic sequences and the changing of DNA. So wouldn't that actually pass down to offspring?
So the issue is, can epigenomic changes be passed down to offspring? And again, if they arose somatically, the answer is no, if they arose in your adult tissues. But there's no question there are epigenetic effects that do get passed on to offspring. And so identical twins are not exactly identical because of epigenetic effects. We clearly have a ringer in the audience here. Okay. So, yes?
Is it black and white where a gene stops and starts, and if so, how do you know where that is?
Is it black and white where a gene starts and stops? And the answer is sort of. So where the coding sequences are, the things that code for RNA that lead to protein, most of that we can figure out where it starts and stops. Now, those start and stop parts can change in different cell types. So one cell you might use one start, and another cell you might use a different one, and another's same with the stop. It can vary from cell type to cell type, but you can map those. So you can actually figure out where they start and stop at different types. The part that's hard is these regulatory elements that I mentioned earlier. They haven't been well mapped. Those are controlling the gene, and so that's the part that's been very, very difficult to pin down. And they can often lie large distances away from the actual structural part of the gene, the part that encodes the RNA. So it's a great question, though. Yes?
Is there any progress in fixing the defect in the genome itself?
No. Is there progress in fixing a defect in the genome itself? So the answer is for certain diseases, there has been good success. Gene therapy has been a real roller coaster ride for the sciences, and for all kinds of reasons. There's been some amazing successes, something called ADA, a particular deficiency that has had, looks like some pretty good success. There was a case where they thought they had success for people who had lost their immune system because they were missing a particular gene, and they added this gene back. This is one of the biggest hallmarks of gene therapy, only to discover when they added back the gene, that preferentially landed in many of these kids. There were like 11 kids, I think, tried, or 15, something like that. And 4 of them developed the gene, the corrective gene landed in a spot that actually caused cancer. So they got cured of the immune thing, but then they got cancer. And nobody could have foreseen that at the time, although now I think there should be a strategy to avoid it, but you have to go all the way back to the beginning. There's been other situations where some of the vectors they use for transferring genes weren't as safe as people thought, and that's actually led to people dying and things not being managed as properly, and those have been huge setbacks for the field. So it's been this kind of thing. So gene therapy, just I guess I'll leave you with one comment, it's a therapy of last resort. It's the therapy you use if you can't do anything else, okay? Okay, one last question, because I want to cover a really fun story I think you guys will want to hear. Go ahead.
So we have identical twins, and they went through puberty about 6 months different. And they're microbiologists, so they tested their own DNA, so they know they're identical. But as a result, one is a little taller than the other. One weighed about 10 pounds more than the other, and went through puberty about 6 months later.
Okay, so the comment is she has identical twins that aren't exactly identical. One went through puberty 6 months before the other. One is taller and, I don't know, same individual weighs more than the other one. Yes, they're not identical. I'm sure there are plenty of scientists who would love to study your kids there. Okay. So okay, let me move on to, I thought I threw up this issue. It's a very interesting story about genetic discrimination. And this has been a huge concern there's the issue about genetic privacy. So yes, you may want to get your genome sequenced, but you may not want it out there. Maybe you want to store it in your own bank. To me, it's obvious that the person who owns your sequence is you. And if you want to disclose it and put it out there, that's up to you. And if you don't, that should also be up to you. But there are big issues here associated. So, these are some of the issues about. So discrimination, I mentioned the misinterpretation that usually is these complex markers. So I want to tell you about a very interesting story about genetic discrimination. It's the most visible one I know of. And it starts with, there were a series of basketball players. You know, basketball is a very stressful sport. And there's been over the years at least 3 individuals who have died of a disease called hypertrophic cardiomyopathy. Perhaps the most famous is a guy named Reggie Lewis who played with the Celtics. He was just, starting to blossom and become a star for the Celtics. And he died in a practice on a basketball court. And afterwards they tested him. But he did have heart problems before that. I think they did clear him. I don't know all the details. But basically, this condition is such that if you have hypercardiomyopathy and you do excessive exertion, physical activity, it's potentially fatal. And that was true for this particular individual. So along came another budding star named Eddie Curry, who played for the Chicago Bulls. And he developed arrhythmia, and the Bulls basically said, you either get this test for HCM or you're benched. Okay? So Eddie Curry decided not to take the test. So the Bulls traded him to the Knicks, and the last I knew he was the second leading scorer on the Knicks. Big rebounder too. And he obviously refused to take the test because if he were tested positive, he felt it would have ended his career. So this is certainly genetic discrimination, right? If he doesn't take the test, he's off the team. And that did happen. So the big issue is then, should an employer be allowed to require an employee to take a genetic test? And should Eddie Curry have the test done? So you can make your own judgment here. My own view, it's interesting, I presented this to an audience in New Haven to see what people thought. And I said, how many think that Eddie Curry should have the test done? Most people raised their hand and said yes, and there were a few people who said, no way, basketball is his life, man, you can't do that. So my own view is Eddie Curry should have had the test done. Should an employee be allowed to require an employee, should an employer be required to have an employee required to take the test? And it's an interesting question, right? So you have to think about it a minute. To me, the answer is that, again, you shouldn't require, but they probably should have. You know why they did it, of course. They didn't want him to go out on the basketball court and die and therefore be liable. So to me, the obvious thing would have been to have him sign a waiver or something like that. But I had to think about that. I didn't sit there like you guys and try and do it off the cuff. But you can see, so I think most issues about genetic discrimination are obvious, but there are a few gray areas. You know, how about airline pilots and narcolepsy and things like that? So you could envision a few scenarios that might be the case. So, but for the most part, it's pretty obvious. I think you know, employers, in my mind, shouldn't be allowed to do this. So, but nonetheless, so one is the employer, and then the other is this health insurance. And so, recently, a law has been passed in Congress that you can no longer discriminate based on employment or based on health insurance. And so, that is the law. So that just passed recently. Now, does that mean it doesn't happen? I don't know, right? So just because something's a law doesn't mean it doesn't happen. But one thing that was not covered is, in fact, life insurance. So if you think about it, that's pretty relevant. If a life insurance company were to ask you whether you had certain conditions and you had disclosed in the public that you, in fact, had these, well, you could actually perhaps not be considered. So there's still, it's not the kind of non-discrimination act that I would like to see, but it's a big step forward anyway. Oh, good, we have 5 minutes. So I do want to just let you know that, you know, at Stanford, and one of the reasons I came here is that we really want to be leaders in this whole area in virtually all aspects I've described. And so I'll give a little selling point for what we're trying to do here, and feel free to make comments either as we talk, or maybe at the end I can certainly wait outside or something. So we are launching a new center for genomics and personalized medicine, because I am a believer that anyone, as I said before, anyone who wants to get their genome sequence will. And I can envision a time, I'm not saying it's going to happen, but I can envision a time where kids are sequenced at birth. You'd certainly get genetic tests at birth now for PKU and certain things. And if there weren't discrimination, I could envision a time when that might happen. And if you think about it, there are actually ways in which you could get a genome sequenced from a kid before birth, because some of the fetal cells are circulating in the mom, and you could actually fish those out and in fact sequence genomes. And this isn't science fiction. This is, hasn't been done, but it's not so far away. You can do genetic tests in the mom for the kid, okay? So this will be coming down the line. It raises a whole series of issues. But these are issues that we need to face. Just because they're serious doesn't mean we should ignore them. It means we have to deal with them and decide what responsible policy is. So this is coming. As I like to say, the train has left the station. You know, there are lots of people who are thinking, well, how do we manage this? Let's put everything on hold. It's not going to be on hold. People are going to charge ahead. You know, individual liberties are strong in this country. Who's to say I can't get my genome sequenced, right? That would be discrimination. I don't want somebody to tell me I can't get my genome sequenced. So those are issues. So what do we want to do at the center? We want to, Stanford has a lot of amazing strengths. It's got very strong technology development. We have people who know how to apply this. We want to make very key discoveries of gene sorts of things and therapeutics. And we want to move them into the clinic as quickly as possible. And I envision this as a major goal of the center, to go right from discovery to the clinic and keep developing new technologies to push the envelope. We have a lot of, as I say, strong faculty to do it. We're uniquely suited because there's a lot of really very strong and innovative companies in the whole Bay Area, many of whom are eager to work with us to try and achieve this goal. So I think this would be an incredible thing to try and accomplish here. And it's something, as I say, that's drawn me here to be able to pursue this. There are going to be many aspects of the center. There's going to be genome sequencing. There'll be issues about how to interface it with the clinic. There'll be issues about the ethics, and there'll be issues about education. Our doctors of tomorrow have to be able to deal with everything I just told you. I mean, again, people are going to come up and ask about genetic tests, and if our doctors don't know how to do this, we haven't trained the right people, or we haven't trained them right, I should say, would be a better comment. So the point is we have to change the way we educate the future doctors in this country. So all of them have to have a reasonable amount of genetic competency and going forward. So one big issue, one big thing we're doing is launching the sequencing center. It's already moving. These machines we're bought some of these aluminum machines, and we have discussions underway with a number of other companies, including 454. And this is not cheap stuff. These machines, I didn't say, but they cost $500,000 to $1 million. Okay, so, and then you pay $50,000 to get a sequence on top of that. So it's very, very expensive things, but we think the payoffs are huge. And the prices are going to drop, and there are more companies coming down the pipeline. Pacific Bioscience, I've listed here. I haven't mentioned them before. They're clearly one of the promising technologies that will be out very soon. The other thing is, it gets back to this issue, our genetic predictive value is quite poor in many cases. And we want to try and bring other kinds of information in. For example, maybe understanding the sequence of a person when combined with other sorts of information, like blood tests. I think the blood tests we do now are not at all going to be like the blood tests in the future. We look at a few things, tens of things. We should be looking at thousands of things to really understand what's going on. And we should do it with something that looks like a cell phone. Okay, so you can do tests, and maybe you'll want to do it yourself. That would be the real future, so that you can quickly, with the combination of genetics, along with really comprehensive tests, try and understand what's going on. So if you're not feeling well that day, you run one of these tests and look at 5,000 proteins and say, gosh darn it, I've got swine flu. You get the idea. So I think we can combine other kinds of information. The way we're doing it is just, it's caveman compared to what it'll be, I think, 10 years from now. And so the goal at the end of the day will be to combine different kinds of information. Genomics, I've indicated up here. Proteomics, where I haven't gotten into this, but where you look at thousands of proteins and try and predict so-called normal from disease states here. And I'll just leave because we're out of time. We want to look at lots of different disease areas as part of the center. Certainly all the major diseases, and this is by no means a comprehensive list. This is just a subset of the things we'd like to look at. I'm sure you can probably think of your own favorite disease, and there's people here who are very interested in it. So I guess thank you very much for having me here.
So I think you can see why we recruited Dr. Snyder here. And he has graciously said that he's going to stay after and answer some more questions. Next week, you're going to have Dr. Julie Theriault. She's going to be coming into the microbe world. Please read in your text so you're ready for that, and we'll see you next week.
For more, please visit us at stanford.edu.